All right, Bio 30, here we go. Karyotypes. So there is actually really no place to put this in your notes. So you're just going to take a few notes on the side. You're going to need this for your assignment that's coming up. So please pay attention and follow along. A karyotype. That is not a made-up word. Instead, it is a word that means mapping out your chromosomes. As you can see in the video here, I have some colored ones here. And here they are all lined up. So chromosomes, karyotypes, how do we do it? Well, they're mapped out according to size, banding pattern, and the centromere position, which is essentially the little section in the center. Please write that down. You're going to need to know that. Okay. Now, why do we need to use karyotypes? Well, karyotypes are really important to help detect chromosomal abnormalities such as trisomies and monosomies. That was chromosomal mutations, which we learned yesterday. Okay. We can do a karyotype and typically around the 3rd month of pregnancy. We would have learned about the types of techniques to do this in chapter 14 and 15. We did not cover them, and that is okay because I'll recover them right now. So a karyotype, we have to prepare them. So what is being done? Well, essentially, you have to take blood from the person that you are doing the karyotype on. So you're going to take the blood cells, you're going to centrifuge it. Centrifuge is just a really fancy word for spinning it really fast and separating out the pieces. So the white blood cells are transferred and treated to stop cell division, and they stop them usually around metaphase is usually where they kind of stop the process. Okay. And so what happens is then they're going to take the sample, they're going to stain the sample, and then they're going to look at them and they're going to map them. Now, this used to be done by hand. Now we can take a photograph of it and use computers to arrange it. So understanding how to prepare a karyotype is important. So stop here and write these few stages down because it is important to understand what is being done to figure out a karyotype. So obtaining the cells for a karyotype, there's a couple options you have. Again, we would have covered these in 14 and 15, the human reproduction, but we skipped them because I knew I was coming back to them here. So there's a couple times. Chorionic villi sampling, CVS, is one of the very first ones that can be done. It can be done as early as 8 weeks of pregnancy because remember, the chorion is only around for so long. By the week 12, it's turned into the placenta. So if you're going to go and to do chorionic villi sampling, it's done really early. So they use an ultrasound to find fetal position. Then, as you can see here, so that's an ultrasound here, then they go up through the vagina into the cervix and into the uterus and take some of the chorion. So take little pieces of the chorionic cells and they pull that out and use that for the sampling. The other one you can do is something called an amniocentesis. This happens usually around 12 to 15 weeks because they're actually taking placental, like they're going in and taking amniotic fluid after the placenta has fully formed. So the ultrasound, again, you go on the outside, you find fetal position, and then you're going to use a really, really long needle that kind of goes actually near the belly button, and you go in and you withdraw fluid. The fluid that is found around the fetus in the uterus, the amniotic fluid, actually has fetal cells sloughed off into it. So you find those fetal cells and you analyze those fetal cells. The reason you're doing this is because you're concerned about a chromosomal abnormality. Maybe someone has something like Tay-Sachs or hemophilia or color blindness, which can all be genetically passed on. Maybe you're checking to see if your child is a carrier or is affected. You can also say the same for cystic fibrosis. So once you get the karyotype, the idea here is that we have 46 chromosomes. They make pairs of 23. So a karyotype is all about matching them together. So humans have 23 pairs of chromosomes, females 23 homologous, males 22 with a pair of sex chromosomes. So what you're going to do is you're going to sit there and you're going to look through all your chromosomes and try and match them so that you have a beautiful little picture that looks something like this. Now, if we were to look at it, this is more realistic to what you would see. You would have a whole set of DNA chromosomes right here. They would all be mixed together and you're going through and you're trying to match them. So when you match them, you actually have one from mom and one from dad. 23 from each parent, 46 in total. And at the very end, you can figure out if it is male or female. Remembering the X chromosome will be much larger than the Y chromosome. So if this was a male, this would be half the size or even a quarter of the size. So that is a karyotype, and it is used to try and help us distinguish if there is non-disjunction that has occurred, because you can look for certain ones like trisomy 21 or trisomy 13 or trisomy 18 or Klinefelter syndrome, which is trisomy 23. You might have Turner syndrome, which is monosomy of 23. Okay, these are all different things that we can look for in a karyotype. Today's activity, you're going to go online and you're going to do one of these on the computer. If you are struggling with being able to do this on the computer, you have to allow your computer to run Flash or Adobe. If you don't have it, I will have a copy of the paper copy in the Google Classroom. You can screen capture and try to move them around by taking snapshots and trying to place them together. I will also be posting the our very last assignment, which starts with a karyotype, and you need to do some cutting out. So I'm giving you the weekend to do the cutting. So please make sure that you print off your karyotypes to cut out. If you cannot print, please let me know and we will figure something out. All right, ladies and gentlemen, that is a karyotype, and I hope you have a great weekend.